My name is Cynthia Magro. I'll be discussing our BGD paper, the different pathophysiologies that underlie COVID-19 perniosis and COVID-19 thrombotic retiform purpura. There are just two distinct acral presentations of COVID-19, perniosis, which we see in mildly symptomatic patients, typically children, and thrombotic retiform purpura, seen in severely ill patients with acute respiratory distress syndrome. The goal of the paper is to really demonstrate the disparate pathophysiologies that underlie these two forms of acral COVID-19, utilizing formalin fixed paraembedded tissue, whereby we had biopsies from patients with COVID-19 perniosis, idiopathic perniosis, and thrombotic retiform purpura. The samples were explored for inflammation, interferon signaling using the immunohistochemical stain Microvascular protein, microvascular complex deposition, IL-6, SARS-CoV-2 protein, as well as RNA expression. So what were the main findings? In COVID-19 perniosis, there was a robust inflammatory self-infiltrated superficial and deep, with marked MXA positivity indicative of interferon signal, in contradistinction to thrombotic retiform purpura with no inflammation and a negative MXA staining pattern indicative of a depleted or absent interferon signal. The infiltrated perniosis was a T cell and histiocytic one with a minimal B cell response. There was no evidence of complement-mediated microvascular injury, and the SARS-CoV protein, as well as RNA, was minimally expressed only in a few extravascular mononuclear cells, as was IL-6. In contrast, in thrombotic retiform purpura, there was no inflammation, microvascular thrombosis, negative MXA staining results, extensive SARS-CoV protein in the microvasculature, but paradoxically in the absence of any viral replication indicative of Pseudovarions, which were nevertheless capable of activating the complement pathway and also upregulating IL-6 and caspase-3 expression in the endothelium. The source of the Pseudovarions is the extensive viral replication occurring in the septal microvasculature of these patients. So why is this study important for dermatologists and patients? Patients with COVID-19 perniosis demonstrate a robust interferon-driven response, leading to an influx of T cells and monocytes that wipe out the virus and lead to mild disease. At the same time, this inflammatory self-infiltrate could have its consequences in terms of a multi-organ inflammatory process. Patients with thrombotic retiform purpura have minimal interferon signaling, excessive viral replication in the lung, whereby the infected endothelial cells release Pseudovarions into the circulation, which dock to ACE2 positive microvascular beds like the skin, activating the mannan-binding lectin pathway, which in turn amplifies the alternative pathway and the coagulation pathway, leading to distant microvascular injury and thrombosis. Thank you.